Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Bioassay Method Transfer Strategies to Reduce Variability. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, note that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Bioagilytics, who developed the content for this presentation. Bioagilytics is the trusted partner for bioanalytical services throughout every phase of the drug development process. As a leading global bioanalytical laboratory, the company provides established and emerging pharmaceutical and biotechnology organizations with pharmacokinetic, immunogenicity, biomarker, and CMC analytical testing services in a GXP environment. Now I would like to introduce our moderator for today's event. Jessica Bridges Weaver brings over 20 years of expertise in pharmaceutical development of upstream and downstream processes and bioanalytical testing in support of chemistry, manufacturing, and controls. Before joining the science office, she led a team of CMC scientists as an associate director at Bioagilytics. So now without further ado, I'd like to go ahead and hand the presentation over to our moderator for today's event. Jessica Weaver, you may begin when you're ready.
Thank you, Ryan, for that introduction. Today, Bioagilytics would like to present how we as a CRO handle assay transfers, specifically focusing on the complex bioassay or potency assay. And before we turn it over to our panel of speakers, let me introduce you to Bioagilytics. Bioagilytics is a comprehensive CRO. We're leading the industry in many statistics. Our scientists are our IP, and over half of the company holds master's degrees or doctoral degrees. We have over 1,100 employees to date globally, and that workforce annually supports over 800 assays to be developed, as well as processing over 1 million samples in a year. We do have a global footprint. We have a team that spans across the globe, as you can see. Today, our speakers are part of our CMC team, and those are highlighted in yellow. They're representing the Durham, North Carolina headquarters, as well as our Hamburg, Germany, European headquarters. At Bioagilytics, we approach our relationships with our clients as an extension of their laboratories, and we can support them from discovery phase all the way through post-market approval with life cycle management of their molecule. Bioagilytics is a very diverse CRO with experience spanning multiple different modalities, and we have supported over 60 FDA approvals since 2021. Ryan, I think I'll turn it over to you for the first polling question.
Yes, thank you so much. So appearing on everyone's screen right now is our polling question. You can participate by selecting on any of the answers that you see in front of you and then clicking submit. The question that we're asking is, does your team or company outsource CMC analytical testing? Your answer options are yes, all spec release and characterization assays, yes, cell-based potency only, yes, but only safety and compendial testing, or no, we perform all testing internally. We'll give everyone some time to participate in this question. We appreciate you taking the time to do so. Again, select any of the answers you see in front of you and click submit to register your vote to the question of, does your team or company outsource CMC analytical testing? I'm going to give you all a little bit more time to consider an answer and participate, and then we'll hand it back to our speaker to continue the presentation. You're all doing great submitting some answers. Thank you very much. A little bit more time, and then we'll see where the results are. Okay, looks like we have most of our results in. Let's go ahead and see how it came out. It looks like we've got 10% of you selecting for yes, all spec release and characterization assays, 10% then also for yes, but only safety compendial testing, and then 80% no, that you perform all testing internally. Thanks a lot again, and I'll hand it back to our speaker.
Thank you, Ryan. All right. Thank you all for those responses. And as we move past an introduction to bioagiletics, let's go into the presentation on assay transfers. We have a few considerations we hope you walk away with today. We want to consider the different phase your assay may be in during transfer and what the guidance may advise you for that. We are going to specifically speak to different regulatory guidances on transferring to and from sending and receiving laboratories. We do have some case studies that the scientists will speak about today that they are managing themselves. And then also we will look at understanding phase appropriate method transfer considerations, comprehending the FDA, EMA, and where possibly if you're transferring across those regulatory agencies, the gaps may be, and applying those strategies to overcome any common bioassay transfer challenges. With that, I'm going to introduce our speakers that we have on the panel today one by one. The first up, we're going to have Lee Lawden. She holds a PhD from NC State in biomedical, the PhD is from biomedical engineering from Duke University. Her bachelor's degree was from NC State in biomedical engineering as well. She has been with Bioagiletics, I believe, now for almost 6 years. She has worked diligently in the background developing multiple different CMC assays, primarily potency, and she has extensive research experience in the range of cell lines and drug development. She also works not only in our cell-based potency area with ELISAs, but also PCR platforms. Our second speaker today is going to be Katie Harcher. She holds a master's degree. Thank you, Katie, for coming on camera. She comes to us from pharmaceutical companies. She's used to using CROs, and she joined us as a CRO scientist herself. She now leads an analytical development team here within Bioagiletics, and they perform everything from ELISA, MSD, flow, cytotoxicity assays, proliferation assays, PCR-based. And her degrees came from John Carroll University as well as the University of Virginia. Thank you for joining us today, Katie. And our final speaker today is going to be Julia Hecker. She represents our Hamburg, Germany lab, and she's a principal investigator at our headquarters there. She manages CMC projects for us in Hamburg. She also works very diligently with the other speakers on the call for assay transfers as we're transferring internally from our Durham headquarters to our Hamburg headquarters. She has over 10 years of experience prior to joining Bioagiletics at a different CDMO in Europe. She came to us to head up specifically our bioassay team there in Hamburg. She works across multiple different modalities and MOAs. She's a specialist in cell-based assays. You may have seen her speak before on flow assays as well, but she also is very familiar with assay transfers and a good resource for that. So with that, I'm going to turn the podium over to Lee Lawden to kick us off.
Hi, everyone. I will be starting the presentation with an overview of the regulatory documents that are primarily involved in method transfer. So moving to the first slide to cover these main regulatory documents. The first and foremost is USP 1224, and this is the primary regulatory document that we look to in terms of method transfer process. It involves the guidance for outlining the process and requirements to qualify a lab for transfer of a variety of analytical procedures. And with this document, we can ensure that there's consistency and accuracy of our execution during the transfer process between sending and receiving labs. Now, also some key documents that are involved are the ICHQ2-R2, which involves the validation of analytical procedures. During your method transfer process, you may want to do some sort of validation, and this document will allow us to understand the key parameters that we want to test in validation. Also, transfer processes can be international, and in terms of the European documents, the EU GMP Chapter 6 outlines the guidelines for quality control of analytical procedures in the EU. Moving on to a more in-depth discussion of USP 1224. So like I said, this is the document that outlines the process to qualify a receiving lab for the use of an analytical test from a sending laboratory. And this can occur under non-regulated or regulated conditions, but there are a variety of types of transfer processes that we usually encounter. So you could have an interlaboratory or departmental transfer. This could be within the same company, even within the same building. And this may just allow you to maybe you built a new facility within the same company and you want to transfer to another lab within that company. Another option is between sites. So within the same company, you might have other sites. Like I said, it could be difference between the US and the EU. So we could have domestic or international transfers within the same company. Another classic method transfer is between companies. So I'm sure a lot of you are familiar with the developing lab sending it out to a CRO or CDMO in order to transfer their method to and outsource it to another facility to do a lot of the stability testing. Another less common and less known method transfer would be across devices. Sometimes we have transitions between different devices, say your plate reader, you may want to use one versus another. Maybe one is being outsourced and you need to transfer it to a different device, and you'll have to do a method transfer process for that. So moving on to more discussion on the different types of method transfers. There are four main types that we usually encounter. There is first comparative testing, which may be due to just testing a set of samples at the sending site and the receiving site. This may be due to the assay is at a initial phase and you're not ready for validation yet, so you just want to do a comparative test between the two sites to make sure that the transfer has been successful. Another option is co-validation. So if your assay is ready to maybe move up to from an early phase to a late phase, you may do a co-validation at both sites so that you can validate it for a later phase in the sending lab and then also validate it at the receiving lab. So both sites will be involved in the validation process. If the sending lab is not necessarily ready for a validation, you may just do a revalidation, or if the sending lab has already done the validation, you could do a revalidation or partial validation at the receiving lab, and these results will be independent at the receiving lab. However, they can be compared to a previous validation from the sending lab. Another less common and less desired option is the transfer waiver. This is not normal for a complex assay such as a bioassay or any other cell-based assay, but if you are doing some sort of compendial testing where the protocols are very basic and both labs have individuals trained in that platform, you could opt for a transfer waiver to waive the wet lab work portion of the transfer process. So moving on to the transfer itself. Like I said, we have regulatory documents that guide us in how to perform the method transfer, and there are a variety of themes that are pretty typical across all the agencies, and the process is broken down in the basic steps that you see here. So we start kind of with a paperwork exercise, doing a method validation check and a gap analysis. So reviewing the method itself, the protocol, and kind of pinpointing any risks that we see in the transfer process based on historical data that we've already acquired on that method. Then we move on to evaluating training of the individuals involved at sending and receiving sites to assess any risks there, design a transfer plan and acceptance criteria based on historical data. We execute that plan and then analyze the data and assess any post execution deviations or risks that we've had. And all of this is documented finally in a transfer report that allows us to assure that the process was completed accurately and that the method has been transferred successfully. Moving on more towards these risk-based analysis. So like I said, we start with this paperwork exercise and a risk-based analysis of the method. We look at the method itself and any data that's historically been accumulated. We look at maybe any risks in terms of how the assay has performed in the sending lab's hands, any specs or acceptance criteria that we've already set, and try to pinpoint any risks that we might ensure. And then also looking at the two laboratories, we want to make sure that both labs have experience with the assay and if there's any relationship between the two labs, and if we can foster that relationship, because as we all know, assays transfer better when the scientists can communicate directly with each other. And this risk-based approach is very useful for saving time and money, which we know is crucial in the drug development pipeline. So moving on more towards this risk-based approach, Like I said, there are a variety of factors that we want to look into. We first look at the people. We make sure that they're all trained. If there is not adequate training, we want to maybe foster that from the sending lab to help get the receiving lab trained. We look at the equipment. We want to make sure that the equipment that we're using from the original method is the same at the receiving site. If it's not, we want to look at the risks there, in particular the software, because sometimes the way certain softwares analyze the data could have an impact on the results. Also, we want to look at the reagents and materials, particularly critical reagents. I've mentioned that you can do a transfer internationally, and sometimes the critical reagents and materials that are available in one location may not be available by approved vendors at the other location. We also want to look at the lab environment, if there's any changes. This has less of an impact, but maybe different labs have different setups that may have a minor impact on the data output. Most importantly, we want to look at the method and protocol and instructions. We want to make sure that we have all documents and SOPs associated with the execution of the method to make sure that it is transferred and executed exactly the same at the receiving site as at the sending site. So most of this has been more of a paperwork exercise, like I said, but we want to also do the actual wet lab work. So I will hand this over to Katie now, and she will discuss that aspect.
Great, thank you, Lee. So like Lee mentioned, moving into more of the wet lab transfer steps, first we'd look at training. So thorough training of the new individuals at the site is critical, of course, to get a successful assay transfer. One of the key things you can do is utilize the same material and equipment whenever possible between the two sites. In doing so, you're limiting variables that may cause discrepancies during the training phase. Therefore, what you're allowing is more of the focus to really be on the method execution instead of looking at things like different materials or equipment. As we all know, often method SOPs lack a bit of precise defined detail and key assay steps. And sometimes these nuances are lost during an assay transfer. So for example, you can think there may be a specific pipetting technique where you have to use either forward or reverse pipetting, and it's very specific for a critical step. Another example would be if there's a specific location to place an assay plate within the incubator to get best results. And so it's important that these key tips and tricks get captured from the initiating lab and get transferred to the new trainees at the new site. This can be especially important because occasionally there are limited personnel who've worked on the method at the original site, and so that knowledge is really just with a few individuals and can be lost if it's not captured and transferred to the new site. So having those individuals available for questions or even better, to come on site and do a training visit can be very useful in a smooth training during this transfer phase. So beyond this, we will consider what is needed for the transfer plan. Okay, so for a transfer plan is very critical and crucial in order to ensure regulatory compliance, data integrity, and a clear understanding of what needs to happen at each site to have a successful assay transfer. To put this in place, the first thing that you should do is consider the scope and the objectives of the assay transfer. Determine what type of transfer needs to occur, just like Lee mentioned. Is it a co-validation? Is it a partial validation? Is it comparative testing? Most likely not a transfer waiver, but that certainly should be considered as well. Once you've defined the scope and the objectives of the transfer, now you define all the details that go into that transfer. So some of these, of course, include which method will be used, the tests to be performed, the materials to be used, and it's important to define the lots of the materials because these may be either the same or different between sites. The acceptance criteria both for the actual method and for the assay transfer needs to be defined. This criteria needs to be defined prior to starting any lab work. That's very important. How the data between sites, if we're comparing data between sites, will be compared. It's important to define the number of participants, the types of machines, and the number of days and assays that will be run to do the transfer. Another important consideration is whether you'll be using real samples to do the assay transfer testing, or whether you'll be using a control sample such as an assay controller or reference standard. Once all of the transfer plan is put in place, We now need to consider, or the details for the transfer plan are put in place, we need to consider the level of rigor and the needs based on the level of the phase of the drug to be transferred. So for an early phase transfer, you may or may not have an assay that's already been qualified or validated at the previous lab. If the assay has not been qualified or validated, you may just need to implement the new method at the receiving lab. As mentioned, this would you would probably be comparing to historical data or development data at this case from the new site to the old site, but you wouldn't have a validation report or qualification report which you could use for comparisons. If the method has already been qualified or validated at the previous site, the new site at minimum needs to assess accuracy across a limited range, perhaps just at the 100% test level, and precision. In some cases, it may be suitable to expand the parameters that you'd look at for this type of transfer, looking at selectivity, specificity, repeatability, maybe system suitability as well. But these will depend on what was done at the previous site and, again, the risk assessment that was done during the paper exercise for the assay transfer. Another thing to consider is if changes have been made at the site, at the original site prior to the initial validation being performed. So if there's any changes such as changes in cell-seeding density, changes in the way the drug dilutions are made, this could expand what you need to do for the assay transfer step. At phase 2, you certainly have to do a more robust transfer. What we normally do at phase 2 would be look at parameters such as linearity, accuracy at multiple levels. 3 is fairly typical, so you don't have to go too far beyond that. Range and intermediate precision. Sometimes limited robustness is included, but again, this really depends what was performed at the original site and what the risk assessment says in terms of what is needed to show that the assay method performs as expected at the receiving site. It is important, though, to look at multiple operators, days, and pivotal equipment, such as the data collection instrument when you're doing your assay transfer. Now, moving on to phase 3 in commercial, your assay transfer validation will be more like a typical assay validation. So in this case, you will include all the things we talked about for phase 2, but you'll expand upon these. Particularly for accuracy, you're probably going to include more levels than you did at the phase 2 type assay transfer. So perhaps 5 levels is more of a minimum range that you would need for accuracy assessments. You may also include more extensive robustness. Look at things like specificity, reagent stability, sample stability, and stability indicating properties of the assay to make sure that you can detect degraded samples. So this is more similar to what a full phase 3 validation would look like. Stringency of testing will be increased. You may need more replicates for the testing within the sites and across sites so that you can get a better variability within the sites to compare, so you can assess whether the transfer was successful. You will be aligning your assay acceptance or your assay transfer criteria with the prior validation activities that were already performed or have been performed in the earlier phases. Though if an earlier phase validation has been performed, it's probably stricter at this point with your acceptance criteria. And just to keep in mind, as your drug is, approaching or at this point already approved for commercial, the goal of this transfer process is to show that the transfer does not adversely impact the product quality. Now, as Lee mentioned, though, you do have to balance some practical considerations such as cost and the time it takes to do the assay transfer, because of course those are important for a drug life cycle. So a lot of little nuances you have to keep track of. So I'm going to move now into presenting the first case study. So case study 1 was a multi-site co-validation of a cytotoxicity potency assay using different plate readers and different critical reagents across sites. So for this case study, the drug that was used was an antibody drug conjugate, an ADC. In this case, this drug was used in a cytotoxicity assay, which had already been validated at a phase 3 validation at the primary site. The risk assessment for the assay transfer determined that a multi-site co-validation was needed. Bioagilitics in this case served as the secondary lab for this co-validation. During the transfer risk assessment, we did identify that a critical reagent, in this case FBS, was unable to be imported into the US. The first primary site was in Europe, and the FBS they were using was unable to come to the US for testing. And so since this was a cell-based assay, we knew that there was a risk that changing the FBS could have an impact on the assay. And so we had to look very carefully for a equivalent source of FBS to use in the US. So this was carefully assessed before purchasing the FBS, and it was also assessed before actually beginning the actual assay transfer activities. So during the feasibility stage at the US site, at our site. Another thing that was identified during the risk assessment was a difference in plate readers, which you'll see on the next slide. So you'll see on the left, this is a Synergy Neo 2 plate reader. This is the plate reader that was used at the Bioagilitics site. The plate reader on the right there is a SpectraMax M5. This was the plate reader used at the primary site during the initial validation. While these plate readers both had very similar capabilities and it was determined that they could both be suitable for the assay, there were concerns that there could be impacts on the assay results using two different plate readers. One of the notable differences was some slight differences in the way the detection in machines through the photomultiplier tubes was achieved. So this was something that was carefully investigated, but it was seen that while there was some shifting of the readout values of the data between using the two different plate readers, it didn't impact the overall results from the assay. And so therefore, both plate readers were deemed acceptable for use within the assay. On the next slides, we'll look at the method and the drug. So on this slide, you can see a little schematic here of the antibody drug conjugate, which is the drug that was used for this assay. So the antibody drug conjugate was a monoclonal antibody that was linked to a cytotoxic drug, which I'll call the toxin. What happens here is when the antibody binds to a specific surface receptor on the surface of the cells, it's then the whole ADC is then internalized. And then within the cell, the antibody is cleaved from the toxin. The toxin is now then free and able to exert its cytotoxic effect inside that cell, which leads to cell death. It's important to note that healthy cells do not express the receptor of interest in this case, and so they were minimally impacted by this ADC. The assay itself was utilizing cells that did have the receptor for the ADC. And on the next slide, I'll give you a little more detail into the assay. Okay, so as I mentioned, it was a cytotoxicity assay. It's a cell-based assay. And so the way the assay was performed were a dilution series was made of the antibody drug conjugate, put in the assay plate, and then the target cells were added to that assay plate. The plate was incubated for 3 to 4 days at standard incubation conditions. After the end of the incubation, a detection reagent was added and the data was read on the plate readers. The data was then analyzed by using nonlinear regression, where a 4-parameter logistic curve was fit to both a reference standard and a sample, and then comparisons of the dose response curves for the reference and the sample allowed determination of the relative biological activity of the samples. One interesting thing that was noted for this assay while we were doing this transfer, there were in-depth discussions with the analysts from the primary site where they pointed out some very particular technical steps that we had to pay attention to. So some of these involved some instructions around the cell counts. There were also some very particular pipetting practices at critical steps. And because they were very aware that these had an impact on the assay, the initiating site actually sent videos of themselves performing the assay to our lab, which has been very helpful both during this assay transfer, and those videos are still utilized actually for training of new analysts on the assay to date. So that was a very helpful trick that you can use when you're trying to do an assay transfer that can help for a smooth transition. So moving forward, I'll show you the parameters that were assessed in this co-validation. So in this co-validation, we did the risk assessment and came up with what was needed for the co-validation. And you can see here the different parameters that were assessed during the actual co-validation activities. We looked at linearity across 5 target concentrations, N of 3 at each target concentration. At Bioagilytics, 1 analyst was used. At the primary site, there were multiple analysts used. Precision and reproducibility was also investigated. Accuracy was investigated, and then robustness, which in this case was utilizing a force degraded sample to look for stability of the sample. And you can see on each little box there was clearly defined criteria put in place for the assay. And in this case, one other thing that was important that is not captured right here is that the replicates to be used, which says here that we used N of 3, these were determined to be true replicates. So each individual replicate was prepared from an individual sample. So it was not pseudo replicates or technical replicates, which we may use in other cases and other assays. The reason that this is important to look at is, especially for an activity like this, we determined that this would be a better way to get a look at the true variation within the assay, both inter and intra-laboratory variations. So looking at across the assay plates, across the testing days, across the labs, of course, and across the analysts. So this was an important thing and something that should be defined clearly when you're writing your assay transfer plan. And on the next slide, I'll show you the results. So the results are shown here for the bioagilytics testing. Overall, this assay transfer was successful and the co-validation met all the predefined acceptance criteria for all the labs that were involved. This led to us being able to confirm that the assay performed comparably across the sites. One important thing that was also looked at, in addition to just the predefined acceptance criteria, the data was assessed between sites to look for a bias or a systemic error. So for example, to see if site 1 had all results that were trending low, even though they were within specification, they were still trending low. And this is important because sometimes when you introduce a new instrument, a new analyst that may have, a slightly different technique, a new reagent, as I mentioned, FBS across the sites, you may see a trend to a bias at one of the sites. And it may not look incredibly important at the beginning, but over time that could lead to a divergence in your test results between your sites. And that can be very important when you're using these tests for lot release or stability testing for samples. In this case, we did not see a bias, so that was great, and everything went really well with this co-validation. It was successful. At this point, I'll turn it back over to Lee, and she'll present case study number 2.
Thanks, Katie. All right, I will discuss case study 2, which was a multi-site international co-validation of a cell-based potency assay using different cell counters. So to describe this case study, this was a cell-based potency assay that involved a monoclonal antibody, and it had originally been phase 1 qualified at Bioagilytics in the US. However, the project was moving more towards a later phase, and we wanted to expand the laboratory testing opportunities within the company and decided to move to a co-validation at our US and our EU sites. And this was done at a phase 3 level. So like I said, once you move to phase 3, you can do a full phase 3 validation using your ICH guidelines to determine the parameters you need to test for that level of validation. So this kind of killed a couple birds with multiple stones. So we did our risk-based analysis and identified a few key known challenges that we thought would be problematic during this method assay transfer. So the first challenge was cell counters. Like I said, we can look at different instruments, and I'll discuss this on a later slide. However, during this specific scenario, we had 3 cell counters that were being evaluated just based on the availability at both sites. Another key challenge was cell handling. During the qualification at the US site, it was observed that cell handling was a very specific aspect of the assay, and there were tight acceptance criteria that were put around the assay for cell performance. So ensuring that that same criteria could be met at the EU site was critical for this transfer. Another aspect was the international critical reagent availability. Like I said, sometimes vendors for critical reagents are not the same between different sites between the US and the European countries. So we needed to assess that as well. We needed to make sure that the vendors were qualified, that the reagents were available, and if not, look at the lot availability that we had available and bridge them in comparison to the original assay at the US site. So moving on to more description about the cell counters. So like I said, one of the key challenges was the cell counters that were involved in this assay. The assay had originally been run on the Vicell, but unfortunately the Vicell is no longer being sold by Beckman, and we wanted to expand our options to things that would be available in the US outside of the Vicell. And then also at the European site, we had the Ceilometer K2. So looking at that specific device for use at our EU site, we needed to do a cross comparison between these three instruments. And there were concerns that based on the differences in volume, the mixing, the gating, and the reagents that are involved in all three of these devices, we may see differences in the number of cells that are the cell viability that is counted during the process, and seeing differences between the three instruments. So this could be problematic if your assay is not robust for a range of cell counts in terms of the performance at this range. We needed to assess this during the transfer since it was not originally assessed during the phase 1 qualification. So moving on to more aspects of this method transfer. So like I said, we did a co-validation. The assay was moving from phase 1 into phase 3. So we wanted to do a co-validation at both sites at looking at a variety of phase 3 appropriate parameters, things like precision, accuracy, linearity, specificity, range of the assay, robustness, and reproducibility are pretty classic for a phase 3 validation. And the co-validation design also looked at the risks that were associated and pinpointed early in the method transfer. So we looked to validate it at both sites in the US and the EU, using 2 analysts at each site to ensure that there is reproducibility and precision at both sites as well as between sites, and comparing these 2 new cell counting devices at the US and the EU site in comparison to the original qualification on the Vicell. And based on all of these transfer items, we came up with comparison criteria in the transfer plan in order to ensure that we have precision and accuracy of the method during this transfer process. Moving on, I will discuss how we set this criteria. So looking back at the historical data from the phase 1 qualification, we wanted to assess what had previously been seen and set our criteria for validation appropriately. As you can see, we looked at the precision in terms of percent CV at the range of the assay, as well as the recovery of the relative potency. It's a relative potency assay, so we want to make sure that at each of the NDCs in the range of the assay, we can hit the expected relative potency. So looking at the adjusted relative potency in terms of that recovery was crucial. And then also looking at our references and our controls, there were criteria set around that to ensure that the referencing controls were performing comparably in the sending site and the receiving site. So as you can see in purple, we set validation acceptance criteria that was based off of these qualification results, and the criteria was pretty tight. So we wanted to make sure that we were performing appropriately as the sending site at the receiving site. So if you go to the next slide, I can show the results, and they were very positive results. We had great linearity in terms of the regression lines for the range of the assay. We had a criteria of greater than 0.8 and actually performed with an R-squared greater than 0.9. So this was great. The assay performed comparably in that parameter. Also, the accuracy at all the NDCs and the range of the assay were pretty spot on. The recovery was great. Both sites had between 90 and 110 for the adjusted relative potency, and the CVs were very tight as well, less than 20%. So for a cell-based assay, this is great performance. We also saw repeatability between all of the analysts. The specificity sample and the robustness samples performed as expected, and then forced the forced degradation sample also performed as expected. So this showed that not only can we run this assay, but it's robust and it is stability indicating. It was also very reproducible across all of the analysts, across all sites with a percent CV less than 15%. This is great, and all parameters for this phase 3 validation met the desired criteria. So based on these results, we could say that with great confidence that we had successfully transferred the method between the two sites, and that we could use the new cell counters for the method moving forward. With that, I will hand it over to Julia for the third case study.
Yes, thank you. The third case study is also based on a cell-based assay and a potency assay, but this is read on a different platform. It's a flow cytometric assay, and it measures the phagocytosis. And we compared different flow cytometer platforms to be sure and to really complete our gap analysis for planned co-validation across different sites and across different platforms. Yes, and I'm going to show you on the next slide what this drug is, or what this assay is about. So we used the macrophage cell line, and the macrophages upon application of the drug engulf artificial target cells that are labeled with the fluorophore. And you can see here in the plot of the cells in the cytometer that the really nice population on the right side is getting more granular upon application of the drug, because the target cells are inside the macrophages, and this is leading towards a shift of this population. This assay was transferred internally, and then we prepared for a multi-site transfer. The known challenges of this assay were, of course, the critical reagents. So we had challenges producing large batches of those labeled fluorophores or labeled target cells, which were required to really support the transfer to different sites. We saw that the quality of this product is not the same when it is produced in large batches at our vendors. So we needed to produce this critical reagent in-house and also test the quality in-house. In the beginning, we didn't have much of experience with this assay. We needed to train this assay. And of course, as this is a cytometer assay, we need to set the entire panel at the devices, because in comparison to the plate reader assay, you really have to set the gates and do the compensation and set everything that is required for a flow cytometer assay. And as we move to the next slide, you can see the two cytometer platforms that we are using at Bioagilytics. So normally we always have two devices of each equipment in-house, so that one is having troubles, we can go to the other device so that we don't have issues in measuring samples, and for our ongoing GMP projects. And you can see here the two cytometers that we're using. It's the Fax Lyric from Becton Dickinson and the Beckman Coulter Cytoflex. The Cytoflex is a little bit more convenient in the development, because the software is easier to plan. It is easier to plan your 96 well plates, but they're both can be used in a regulated environment. The software is qualified and is compliant to the regulations. The Lyrics, on the other hand, it was a little bit more faster. So when it comes to high throughput assays, and this is different compared to a plate reader, a cytometer is taking some time to measure the samples. So with a high throughput, the Lyric was also having advantages in our lab. And as we move to the next slide, you can see this assay performing on both of the instruments. First of all, you again see this plots of the cells, how it looked like. Of course, the diagrams are a little bit different that we receive from both platforms. we were able, by gating on the fluorescent marker, to really determine the percentage of phagocytosis that is happening in these macrophages. And this can then be plotted against the dose of drug that is applied. Of course, there were some other gatings before. We gated on living cells, and also we had the cell line marker on this macrophage cell line. But in the end, we really got nice dose response curves that we evaluated in another statistical software, taking the percentages of phagocytosis from the instruments. And you can see from this dose response curves that in the end, they look really comparable. So this is a very specific readout. And although we had to set the panel and get all of the all of the settings on each instrument, we really could get comparable dose response curves. So when you're looking at the samples that we were measuring, we tried reference versus reference in a 100% nominal concentration to see how the accuracy is behaving. And you can see this on the graph on the right side, that across different analysts and then across different platforms, we see very good accuracy and also very good precision. So we see the expected 100% of relative potency on both devices. And we included this in an initial early qualification of the assay that was performed at one site. And as it was requested by our sponsor to be able to, in the later phase of the assay, be able to do release testing in the US and also in the EU. We were planning ahead because this made us confident that we can use the different platforms that we were having at the different sites at that time point to plan for a really large multi-site co-validation. And you can see the parameters that were included in this on the next slide. So all of the ICH required parameters were planned to be included in this or are planned to be included in this co-validation. You can see the validation design, and this time we would span 3 sites, 2 in the US and 1 in the EU. We would include 2 devices in this and 2 analysts per site. And then we of course have to define the comparison criteria. And this is leading to a very complex co-validation, which you can see on the next slide. The design you can see here. So we have the 3 sites, we have 2 analysts, and we have at least at 2 sites the different platforms. And of course, there is a risk connected with this, but it also gives us the possibility that in the end, we can be very flexible in each of the labs because we can use either platform. And we've seen that there are no big differences between the platforms. We could test at either site and we can, we can be flexible which site can test to support the market authorization at the different sites, and also to be able to measure a high throughput of samples. So we are flexible to switch if there are samples that cannot be measured at one site, then they can be sent to the other site to be able to support the large amount of samples that are planned for this. Because this is also connected with the risk. We did kind of a pre-validation to see, because if this fails, then of course you would not have either site validated, but of course there are ways to, in the validation plan or in this transfer plan, to minimize the risk so that you can have either site still validated. But as we wanted to really have comparison criteria in this co-validation, we went on testing, and you can see the results on the next slide. So now we included analysts, we included different sites, we included different platforms, and also we included accuracy samples. So the 3 graphs represent different accuracy samples test at nominal values of 50, 100, and 150%. And always 2 analysts were from the same site, but were using different platforms. So in each graph, we have covered different analysts, different sites, and different platforms. And as you can see, we really get a good recovery over all the different parameters that we've included, and also the precision is really good, and thereby we've minimized the risk. We did an extensive gap analysis so that we can be sure which criteria we have to put in our co-validation plan, and we know that the critical reagent that I mentioned in the beginning, there should be the same lot should be used by all of the sites when we perform this co-validation. And we will, of course, include that testing also in the plan that we're doing at the moment. Yeah, and I think this leads us over to the next poll question, Ryan.
Yes, thank you. So appearing on the screen right now is our next poll question for audiences. So this time what we're asking is, in your experiences, what is the most common challenges you have seen in assay transfers? Your answer options are critical assay procedures are poorly defined, training, equipment or critical reagent differences, lack of regulatory compliance, or other. Please take a moment to consider an answer. Select one from what you see in front of you and then click submit to register your vote. And for those choosing other, if you'd like, we'd even love to hear from you into the chat window what your thoughts are to this question again of, in your experiences, what is the most common challenges you have seen in assay transfers? You're all answering much faster this time. Thank you very much. We're just going to leave it open a little bit longer, though, to try to get as much of you as we can. All right, it's looking like we have most of our answers in. Thank you everyone for participating in this poll. Let's go ahead and take a look at what the results are for this one. It looks like we have 20% choosing critical assay procedures poorly defined, 10% then for training, 50% for equipment or critical reagent differences, 10% of you selecting lack of regulatory compliance, and then 10% for other. So again, thank you very much, and I'll hand it back to our speakers.
Thank you, Ryan. I think this is a very good leading over to the next slide, which is a summary, somehow a summary or a warning of the top 4 errors that can occur when transferring analytical procedures. So as we mentioned in the beginning, there is inadequate or missing documentation. So whenever it's a regulated assay, there is the requirement to have a documented transfer plan. In the best case, this transfer plan is having a gap analysis inside or a risk assessment inside, so from which you can see why you are testing certain things and why you've assessed those as critical things. Yeah, of course, then the acceptance criteria should be defined, and the acceptance criteria should also include how the comparison is done and what of those criteria are criteria that should be compared across the different sites. And then reports have to include, of course, deviations, and the lack of the demonstrated method proficiency is also an error that can occur. Maybe there are things that need to be changed in the method, or there is something that needs, I don't know, needs a different acceptance criteria on the other side, then this has really to be supported in the report and has to be evaluated and well documented. We've already mentioned the lack of training, and that we really like to have hands-on training, because of course it also depends a little bit on the assay and how complicated the assay is, but with every cell-based assay or potency assays, training on site is really something that we recommend. Yeah, the lack of this understanding of the critical parameters, of course, you can communicate, and also the provision of the documents from the transferring site is very important, and this should be performed early on. And this is not covered by training, but it should be the very first part of the 2 sites communicating to each other and talking about this method. The training, in the best case, if it's a regulated transfer, should be documented already. It can also be, so in the transfer plan, it can also be referred to the training why certain things are tested. The suitability of the method, of course, if there are still differences in the equipment or in the software or in the reagents. It might also be small things like lab reagents or so. The best is to really evaluate all of the different reagents and compare site to site, because sometimes the other lab is just purchasing, I don't know, different media or things like that. So really include all of the reagents the method requires. And of course, also the samples that you want to use for the transfer should be should provide enough documentation, and there should be the same samples across the sites. Then there's, of course, some uncontrollable environmental differences that can occur from the different sites. In the end, your data might show poor integrity. Don't ignore any failures. If there are OS events during your transfer, don't ignore them, but investigate probably where do they come from. It can be the case that this also leads to differences or wants to demonstrate differences at the different sites that you did not consider until this time point. And yeah, incomplete data and assay execution, and this of course should also be avoided. And thereby I'm going to have a summary of everything that has been discussed so far. This is really a roadmap of how to proceed for a transfer of a regulated analytical method. So the analytical procedure should be well defined. It should be checked if within your documents or in your dossier it's written the same way that you transfer it to the other side. The roles and responsibilities at both sides should be defined. Then it comes to the already mentioned transfer plan. This one should be phase appropriate and should include, of course, the gap analysis or the risk assessment. The more time is spent on this one, the less problems you will probably have when doing the wet lab work. Training is essential. It should be documented. Best is really on-site and on-hands training. Critical reagents, provide the documentation, provide early on which reagents are critical for the assay, share that knowledge and try to get the same lots for the transfer if anyhow possible. Then the equipment, if there are differences in equipment, evaluate criticality and see if this differences might lead to different results. This can also be included in the gap analysis already. Then the transfer protocol, yeah, as I mentioned before, it should be phase appropriate and it should include all of the above mentioned, and of course the acceptance criteria. Then statistical analysis alignment, how is the comparison performed? What should be compared and how is it compared? Execute the assays and then do the evaluation, do the comparison. Is your data comparable from two sides? If not, of course, include any deviations, include the entire evaluation, why differences might be there and where they might come from. If it can be if it's written down, it can be scientifically argumented, then also this should be performed in the final report. Yeah, include the deviations and any consequences that come from your transfer to the method. And I think thereby I want to close and open. Yeah, of course, would like to acknowledge, of course, and give a thanks to all of our analytical operations team for the great work they do. All of the results that we've seen is produced by them. And then Jeff, our global CMC lead, our director, Shizhan Hu, she's CMC director, our associate directors from the US and the EU, Andrew and Christopher, and of course, all of our sponsors for the very interesting projects that we get and the very interesting work that we can do. And I don't know, Ryan, are there any questions already maybe in the chat? Otherwise, we can open it now.
Yes, thank you so much, all of you, for this very insightful presentation today. As we move into the Q&A session, I would like to direct the audience's attention towards the handouts module, where you'll find some additional documentation related to today's presentation to download for some further reading. I'd also like to invite the audience to keep sending their questions or comments right now for this Q&A portion of the webinar. Now, I've already received some questions from the audience, though, so we'll get ourselves started with those. Looking here, the very first question that I have here just wants to know, what are sort of the top 3 to 4 documents that you as the receiving laboratory would need to be provided by the sending laboratory to perform a method gap analysis? Anyone want to take a shot at this?
Sure, I can take it. I can help answer that one. So some of the documents that we need in order to do the gap analysis and ultimately the transfer are the method that the initiating lab has. If there is a validation or a qualification, any reports that go along with that. Additionally, if there happens to be development reports, which sometimes exist, those are very helpful. It's very helpful to receive any C of A's on the critical reagents, especially things like a reference standard or assay control, but really any of the reagents that are available. Those are some of the most important things that we need to get going with the assessment. If there's a specification that goes along with the assay, that is something else that is needed to be provided for the gap assessment. Those are some. I don't know if anyone else wants to add anything else that I may have missed.
Very good answer.
Yeah. I think that's a great answer for the paperwork that would need to be done.
Moving on to another question here. Someone's just wondering, sort of, how does bioanalytics approach training of analysts on complex assays?
Yeah, maybe. Sorry.
Maybe I can take the US and you can take Julia, or you can take the EU, Julia. All right, so at the US side, we generally follow a protocol where we have an analyst observe and ensure that they watch a trained individual execute the assay the way it's intended. As we all know, some people are visual, so following along with visualizing the assay as well as looking at the protocol itself can give you visual cues on how to execute. Then we have someone observe the individual as they run the assay. Someone who's trained will watch the newly trained person execute after they've observed. And then we tend to do a couple training runs, 1 to 2, maybe more depending on the success of the assay, but just independent execution to ensure that they are able to run it all by themselves. Julia, you want to add anything that you may do at your site?
No, I mean, it's similar. It a little bit depends on the complexity of the assay, but of course, training, on-hand training and practical training is important. We've already done videos of a person doing the assay and shared that across the different continents. But when it's a very complex assay, we prefer to send someone over and do the training on site.
Yeah, absolutely. Julia's right. We've been known, especially in inter-assay transfers from our EU site to our US site, we send scientists back and forth for that hands-on training.
Perfect. All right. Well, thank you very much for that answer, for all the answers and the presentation today. We have reached the end of our time. Now, if we couldn't attend to your questions, know that the team at Bioanalytics will follow up with you, or if you have further questions, you can direct them to the email addresses that are up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Now, additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you, everyone.